Well, hello and welcome back to Science in the Age of COVID-19. Today we are super excited to welcome Dr. Dan Ellis, a research scientist at the Institute for Protein Design at the University of Washington in Seattle. He is in the lab of Neil King, who gave a previous talk in this seminar series. Dan's work has focused on computational protein design and directed evolution, mostly of nanoparticles, cages, proteins that form big multimeric complexes. This has largely been in the context of creating protein formats for nanoparticle vaccine display. And they've worked on several targets, including influenza and, of course, SARS-CoV-2. And the work that he will tell us about today is super exciting, where they have managed to hyper-stabilize SARS-CoV-2 Spike. And he might also tell us about the work on influenza neuraminidase. Okay, Dan, thank you.
Thanks, Lauren. I'm just going to share my screen. All right. Oops. All right. My name is Dan. I'm a research scientist who recently graduated from Neil King's group. And today I'll be talking about some vaccine development that's gone on in the King lab, and mostly focus on stabilization to the SARS-CoV-2 receptor-binding domain and how this can affect vaccine development. So I'll start off with about the first half of my talk describing why our lab is focused on nanoparticle vaccines, and as well the development of an RBD-based nanoparticle vaccine for SARS-CoV-2. And then second half of my talk will be largely focused on stabilization of the RBDs. So nanoparticles are of interest because the immune system in various ways will prioritize immune responses against large objects that have lots of repetitive things on them. So that's kind of a vague statement, but compared to like small molecules or individual macromolecules like proteins or nucleic acids, large assemblies of proteins or other macromolecules are what compose viruses and other pathogens. And many viruses and other pathogens can be studied with a lot of repetitive copies of many different antigens, which are usually comprised of proteins or combinations of proteins and sugars. So because if you were to compare this to like the antigens on their own, individual antigens aren't going to have as much avidity to the B cell surface, which is studied with B cell receptors, while a larger object with lots of different copies of antigens has greater avidity and greater ability to activate B cells by engaging multiple different B cell receptors. There's a lot of other reasons why larger objects get prioritized. Trafficking can play a very large role in that. Larger objects tend to drain better to lymph nodes and therefore lead to better responses as well. So because of this, there's a lot of different vaccine types in the world, and one vaccine type that has validated this philosophy in the clinic are virus-like particles, which are basically just like the outer shells of viruses that do not have all the other viral machinery in them, but just are the part on the outside that the immune system interacts with. Two clinically validated examples of this are human papillomavirus, HPV, and hepatitis B virus, HBV. Both of these are very effective vaccines, and again, are just kind of that outer shell of the virus, which works really well because the virus has a natural architecture that lends to that approach. However, there are lots of different viruses out there that do not have kind of a convenient shell on the outside that you could immediately use as a vaccine. Some examples are membrane enveloped viruses that have like other protein antigens on the outside of them, and being able to build customized vaccines that can kind of use the virus-like particle approach is something that is widely of interest. Now there's been a lot of research over the years at trying to build larger molecular objects to make vaccines. And one large pioneering study is shown here, which was led by Masaru Kaneki at the NIH Vaccine Research Center. Masaru was able to genetically fuse the trimeric hemagglutinin protein known as HA from influenza to this larger octahedral protein assembly called ferritin. And in this case, ferritin as an octahedron has 8 different threefold axes. And by connecting the genes from this threefold symmetric hemagglutinin molecule to ferritin, that leads to the display of 8 different hemagglutinin trimeric spikes on the outside of ferritin. And it all shows that not only is it possible to display antigens on the outside of particles like this, but it is possible to display like native like antigens that really represent what's on the outside of viruses, on the outside of particles. In this study, these nanoparticles displaying HA were compared to the vaccine or a comparable vaccine with the same HA in it, and particularly with the addition of adjuvant, were shown to give much stronger neutralizing responses compared to vaccines, showing potential power of this approach. So there are many other natural self-assembling nanoparticles out there besides ferritin, and ferritin's been used for a large number of different antigens, with one example here showing headless HA antigens to direct responses at more conserved regions in flu. Another nanoparticle that's been widely used is lumazine synthase, which is this natural icosahedral molecule. Its most notable use has involved the display of a designed HIV antigen, EOD, which was recently evaluated in a phase 1 clinical trial. There are other natural assemblies out there. Another one that's been used is encapsulin, which is another bacterial large icosahedral assembly. And there's been lots of different examples out there of these and other natural particles being used for vaccine development. And the ability to just multimerize different antigens onto larger bodies is clearly something that is of wide interest to the field. So where our lab, or the Neil King's group, come in is we are interested in trying to build more customizable nanoparticle scaffolds that could be used for more diverse and tailored purposes. So, so much of the research out there has been using nanoparticle vaccines has been only used naturally existing nanoparticles, and that's made a lot of progress. But you could imagine that if we had the ability to build much more tailored nanoparticles that were built specifically to each purpose, and especially as we kind of gain a greater understanding of the science of how nanoparticle vaccines work, we can start to build really streamlined vaccines that are specifically accustomed to exactly what we want to do and can give the best immune responses possible for each given application. So the King Lab very much uses Rosetta, which is a computational modeling software for protein modeling and design. And one of the main applications that has been developed is the ability to dock different proteins into larger symmetric structures and then redesign their sequences to match those larger structures and favor that exact state. So one example of this is how nanoparticles have been designed in the King Lab, shown here. So say, for example, you have a large symmetric architecture like an icosahedron. Icosahedrons have 20 3-fold axes, 30 2-fold axes, and 12 5-fold axes. So if all using simulations, if you place the 20 trimers on a 3-fold axis and 12 pentamers on the 5-fold axis, and these are all naturally existing protein scaffolds, you can then like move them around while keeping them in an icosahedral conformation and find points where these different components meet and form kind of nice shapes in between them. And then using different algorithms in Rosetta, you can then design novel sequences or mutations into these oligomers, which will then form hydrophobic interfaces or other contacts, which can then drive the nanoparticle assembly. So what's really awesome about this work is that it doesn't only make nanoparticles, but it can make them very accurately. So shown here is work led by Jacob Bale, published in 2016, in which a large number of icosahedral nanoparticles were designed and produced. Shown here are negative stain electron micrographs with the 2D class averages shown on the left and predictions of different 2D class averages along symmetric axes shown on the right, showing good correspondence, as well as some of these were able to be crystallized, and crystal structures matched very, very closely with the design model, showing how this approach is very accurate. Besides being able to make these nanoparticles into a whole bunch of different structures, the ability to make nanoparticles out of two components is particularly of interest because it allows for the nanoparticles to be assembled in whatever situation you want them to in vitro. And in this case, you can purify, for example, the pentameric component separately from the trimeric component, and at a later point mix them together to assemble the nanoparticle. This has various applications, which I'll be talking about throughout this talk. We have streamlined this process by adding stabilizing mutations to many of these components as well, both to help their use in research and to help their translatability and manufacturability for other purposes. So yeah, many of these components do have stabilizing mutations in them to help applicability. So one reason why in vitro assembly can immediately pay off is that it provides some opportunities for vaccine design that are not accessible to natural nanoparticles. One story shown here, which I'll hold off from going into in detail because of other things to talk about today, but we were interested in designing a nanoparticle vaccine that had the same four influenza hemagglutinin molecules that are in the current commercial vaccines. And we were particularly interested in making a version of this vaccine that co-displayed all four of them on the same nanoparticle, which is something we've been investigating for promoting breadth against diverse families of viruses. And what's really advantageous about this in vitro assembly approach is we can control the exact stoichiometry of influenza hemagglutinins that we put into this in vitro assembly reaction and know exactly how much total hemagglutinin of these four different types are in the final molecular product. And we can also make the corresponding cocktail version too by just setting up the in vitro assembly reactions differently. And because we know the exact compositions that's going into this products like this are amenable to manufacturing and therefore amenable to entering clinical trials. This mosaic particle here has been shown in animal studies to give really good results for broad responses against influenza viruses. And this week, the clinical trial is very likely starting for it for phase 1, and it was successfully manufactured over the last couple of years. And yeah, we're very excited to see what this vaccine is going to be able to do in the clinic. So this next I'll be giving an update on some nanoparticle vaccine development for SARS-CoV-2. This is work that I was not immediately involved in, but is work that was led very much by others in Neil King's group and the cores at the Institute for Protein Design, and as well by David Wiesler's lab. That's important to kind of go over the background of this for talking about stabilized receptor-binding domains. So shown here is just kind of a representative overview of a bunch of different techniques that are used for structure-based vaccine development, all using influenza hemagglutinin as a representative antigen. And what is, when this pandemic hit, coronaviruses were much less studied than many other viruses out there. However, there were many pioneers in the field who had studied enough strategies against to generally make vaccines against coronaviruses, that we fortunately were able to get off to a quick start at that. Some groups out there approached it by tackling the native antigen, which in this case was the spike of coronaviruses. There was a lot of effort before the pandemic on conformational stabilization of coronavirus spikes, which became a very important vaccine technique used. And, assuming most people here have been vaccinated with a prefusion stabilized or RNA encoding a prefusion stabilized spike protein. So this had a very large impact on my life, and I hope it has in many of yours as well. And for another approach that had been studied for coronaviruses before has been domain-based approaches, looking at individual domains of coronaviruses. And that is something that I'll be talking about today, because that is the approach that our lab took. So this work here is shown as the production of the nanoparticle vaccine that we developed, which was led by Brooke Fiala in some of our core labs at the Institute for Protein Design. And going into this, there was not many antibodies available to the SARS-CoV-2 spike. But one thing that's generally known about coronaviruses and generally other viruses as well is that if you can elicit responses that block receptor binding, that has a very good chance of eliciting responses that neutralize the virus and therefore lead to good protection. So one advantage of the nanoparticles that we use is that monomeric domains are not that immunogenic on their own, but if you attach a lot of them to individual nanoparticles, that's a very simple and reliable way to increase the immunogenicity of such a domain. So the receptor binding domain alone of the SARS-CoV-2 spike was isolated as a gene and genetically fused to the trimeric component of a nanoparticle called I5350, which when mixed with its complementary B component, will then assemble into a nanoparticle displaying 60 copies of the RBD. This was produced with a bunch of different flexible genetic linkers in between the RBD and the nanoparticle to just explore the stabilities and or immunogenicities of these different linkers, which in the end didn't make too big of a difference. And you can see here by DLS, or dynamic light scattering, that they're all forming the correct size, or the nanoparticles are all forming the correct size, and that similar results are seen by negative stain electron microscopy. So these were next analyzed in animals with a lot of the serology analysis led by Lexi Walls and David Wiesler's group, and then challenge studies performed in Ralph Barrick's lab. And what we see is that after 2 shots, we see very potent neutralizing responses elicited by these nanoparticle vaccines, which compared to human convalescent sera as kind of a general standard from a different organism, are seen to be much, much higher when compared to a decent dose of prefusion stabilized spike, in this case containing just the S2P mutations. We see that 2 shots of the nanoparticles can provide much stronger neutralizing responses. And even a lower dose of like 0.9 micrograms can exceed responses shown by prefusion stabilized spike. These responses were nonetheless protective and showed that the nanoparticles were able to provide very strong protection after the mice were challenged with SARS-CoV-2. Now, a lot of the neutralizing responses that are directed at the receptor binding domain will block receptor binding, which in this case, receptor is ACE2. There are other epitopes that are available on the RBD, which can be protective or neutralizing. And using antibody competition, the Lexi and others were able to show that the nanoparticles were able to elicit responses that hit diverse epitopes on the RBD. So shown here is a binding curve of, say, ACE2 to the RBD on its own. And then as Fabs that are elicited by non-human primates immunized with the vaccine are added in. You can see competition at not only the ACE2 binding site, but other distinct epitopes on the outside of the RBD. So this should, in theory, give neutralization that's focused on multiple parts of the RBD, which is good for helping escape from mutations or preventing escape by different variants. This work was fully further extended in a work led by Prabhu Arunachalam from Bali Palendran's lab at Stanford, in which they studied the responses to this vaccine in non-human primates in the presence of different clinic-ready adjuvants. So in this case, the adjuvants studied include like squalene oil and water emulsions, ASO3, AS37, CPG mixed with alum, and then alum alone. And all here showing that all these different adjuvants can all lead to protective responses, and ASO3 was identified as a clear, strong leader in such responses. So one exciting announcement that happened yesterday is that it looks like we will be securing phase 3 funding to advance this vaccine. So currently this vaccine is being studied by 2 different companies, one SK Biologics in South Korea, and the other is icosahedral here in Seattle. And there's currently 2 different phase 1 and 2 combined clinical studies being performed by SK, with one involving ASO3 and the other involving alum. And we've currently gotten a clearing from CEPI to have funding for the phase 3 clinical trial, which we are very excited about. So how do stabilized RBDs fit in with this vaccine? So this will be a little bit of a longer story to build up to, but I wanted to kind of first share an example of designing stabilized glycoproteins and like what that's normally like. A decent amount of projects that I work on are stabilizing antigens. And antigens are very fragile, and especially when you're like approaching an antigen design problem that has not been studied before. It's a completely different family. It's very hard to, or sometimes it can be very hard to like get a good initial bearing of what to focus on. And because of that, like even when you really diversify the approaches that you're taking and try lots of different types of mutations, you can still have really low success rates. So this is an example from a non-COVID antigen stabilized project, which is influenza neuraminidase that I was working on a little bit. And the this starting antigen had pretty poor stability, and initially tried 32 different designs to just see if they expressed, and a little under a third of them expressed. But when you examine like stability of them, you can the success rate that you can get is just really, really low. And this is something that I've seen and experienced time and time again when starting new antigen design projects. And just to emphasize like how like tricky this can be for finding a bearing on where to start, and particularly that because of this, like any experimental data that can really help guide how stabilizing mutations are identified can be particularly important. So I've been used to doing this in some ways using homology data, but any other like experimental analysis of mutations or other things can be particularly useful. Now, the reason I bring this up is because in the case of the story of stabilized receptor binding domains for SARS-CoV-2, data that was provided by Jesse Bloom's group using deep mutational scanning was a very important part of this story. Led by Tyler Starr and Ali Greeny, Jesse Bloom's group has developed this system in which they display the RBD isolated on yeast and will produce libraries of like a whole bunch of different combinations of mutations or all individual mutations of RBDs displayed on yeast, and do a bunch of different analyses on that. So shown here is data from an initial paper that they published in 2020 on this, which analyzed the effects of all individual mutations to the RBD on expression and on binding to the ACE2 receptor. They have since extended this to many other antigenic analyses, which has been shown just how powerful of a system this is. So kind of going back to the mentality of stabilizing glycoprotein antigens, having data like this that shows what happens if you were to make any individual mutation is just so useful and so important. And you can see from this with the orange representing mutations that hurt expression and the blue helping, that if you were to pick pretty much any mutation at random, the chance that you're going to hurt expression of this thing is high. So being able to kind of figure out where are the spots that we can focus on and which are the traps that we should avoid, and using this data to guide structure-based design is something that I became interested in, and most particularly seeing if this could be used to help vaccine development for our RBD-based nanoparticle vaccines. So what I first did is I looked at a bunch of that data and kind of structurally mapped out where all the different mutations that increased expression of the RBDs displayed on yeast were located. And what was very interesting is that the majority of the mutations that helped expression of the RBD were very much isolated in one region of the RBD. So over here is the receptor binding motif, and the region that these mutations were located in was distal to that and buried. This region has been previously observed in the context of the full spike to be capable of undergoing a backbone shift in which this helix opens up, and as this cavity opens, linoleic acid or just a lipid can bind in there. And this, not shown in this image here, this carboxyl group is kind of capped by a positively charged group on an adjacent RBD. To my knowledge, nobody at this point knows what the function of this is, but it's something that's been observed in a couple different cryo-EM models of the spike at this point. So going back to the context of the isolated RBD, it looks like this region appears to negatively influence the stability of the isolated RBD, and therefore seemed primed to focus on for designing stabilizing mutations. So from the DMS data that was provided to us by the Bloom Lab, I then developed protocols in Rosetta, which is the computational design modeling suite that I was discussing earlier, which will place these mutations into these structures to evaluate which ones work structurally well and favor the kind of closed, more normally seen conformation of this pocket. And as well, not just model individual mutations, but also model combinations of these DMS mutations to figure out which can kind of synergize. And beyond that as well, identify novel mutations or mutations that weren't favored by DMS that could fit in really nicely with these other combinations of really valuable DMS-derived mutations. So shown here is a design that we've called Repack 9, which features this phenylalanine to tryptophan mutation, which helps fill some space. This tyrosine to phenylalanine mutation removes this hydroxyl group and makes a little cavity there. But then this valine to isoleucine mutation will then fill in that cavity to kind of remove this buried polar group, which buried polar groups are very unfavorable, and instead turn that into hydrophobic packing. As well, we also ran some simulations in which Rosetta was allowed to just kind of do whatever it wanted with the space and just generally fill it with hydrophobic packing, which was also explored. And what was really fun about this technique is that because we were leaning so hard on these mutations that were validated and suggested by DMS, we were able to get a really, really high success rate. And every design that we tested showed much higher expression than the wild type RBD. one of the reasons, so we had like a lot of things that were considered successful hits, and narrowing things down would have appeared kind of difficult. One thing that we noticed is that these designs were tested in the context of genetic fusions to the I5350A trimer, which is a component of our nanoparticle. And we noticed that some of the designs had a tendency to form disulfide linked dimers, which is something we'd also seen in the wild type RBD. However, a bunch of the designs did not really form these disulfide linked dimers as much. And we decided to move forward with repack4, this single mutant shown here, and repack9, this triple mutant shown here, for scaled up characterization. So first thing we did is we cloned these mutations both as monomers and already had the genes from them as trimers, which were used for screening earlier. And we expressed them at equal levels of culture and just measured how much yield we could recover. Both the repack4 and the repack9 mutations increased the yield in the context of monomers. The repack4 mutations were seen to not be exactly as helpful in the scale up for the trimers. However, the repack9 mutations did really help the yield a lot for the scale up for the trimers. And so repack9, that triple mutant, very much took the lead as one of our as our leading stabilized candidate. We next tested the thermal stability of these different mutations. So this is what we call nano DSF, or nano differential scanning fluorimetry. Basically what we do is we heat up these molecules over in a very controlled setting and measure how much the tryptophan fluorescence shifts to a slightly different wavelength, with that shift inferring that the tryptophans are becoming exposed and therefore the protein is unfolding. So what we can see here is the repack4 mutations helped provide about 2 degrees of stability in the context of monomers, and the repack9 mutations also provided about 4 degrees of additional stability. In the context of a trimer, the repack9 mutations provided a little over 5 degrees of stability, again showing that this triple mutant and the ability to combine these different mutations was important for finding a leading construct. Next, we analyzed the monomeric RBDs antigenically, and so far I've only done so with an antibody against the receptor-binding motif CV30 and another antibody against a lower part of the RBD in the more core region, which was CR3022. So the ACE2 binding motif was completely antigenically intact. The antigenicity by CR3022 was subtly but not very significantly changed, only showing a really minor change in binding. And it's worth mentioning this is slightly closer to the repack mutations, in all showing that antigenicity was nicely maintained. The next thing we analyzed is the other biophysical features of these molecules, and a really powerful technique for analyzing this is hydrogen deuterium exchange mass spectrometry, with the work here performed by Chengbo Chen and Kelly Lee's group at UW. And basically what this involves doing is exposing the protein to deuterium under deuterated buffers under very controlled time spans, and then comparing, say, the wild type construct to the stabilized constructs and seeing which ones absorbed more deuterium, and particularly looking at that at all these individual regions across this protein, which is the power of mass spectrometry. And more, in this case, more deuterium uptake usually means that a particular region is less ordered or less locally stable. So shown here is a general summary of the stabilized constructs compared to the wild type, showing that the regions surrounding the mutations are becoming more ordered and, in this case, more resistant from deuterium uptake. One example shown here. What was really interesting is that anecdotally, we kind of became noticing that the proteins were becoming less sticky, like they would not stick to like membrane concentrators as much, and there'll be more on that in a little bit. But if you mix the proteins with Cipro orange fluorescence, which is a dye that is gives off more fluorescence when it's in the presence of more hydrophobic things. We found that the wild type RBD gave the greatest signal, meaning that it is the most hydrophobic, and that the addition of more and more stabilizing mutations made this molecule less hydrophobic, which this is surprising because these mutations are buried in the RBD. Between these two pieces of data, our interpretation is that the isolated RBD appears to be or could be breathing or kind of going between those closed and open states, and that the stabilizing mutations are likely locking that down and favoring more of the closed state that prevents like the exposure of that big hydrophobic pocket, and therefore changes the hydrophobicity of the molecule. We did not really expect a lot to happen if we put these mutations in the context of the full spike, but we decided to do that just to see what would happen. And in this case, we used the Hexapro construct developed by Jason McClellan's group, which is SARS-CoV-2 Spike with 6 engineered prolines in that hold it in the pre-fusion conformation. So we also that's this construct shown in green here. And then we also took this construct and added in those repack9 mutations into the RBD in it. The yield that was produced by these was pretty similar, with a little bit more signs of aggregation shown by the repack9 version, showing that it might be a little less stable in the context of the spike. We also performed thermal denaturation with nano-DSF, both in the absence and in the presence of linoleic acid, which is the lipid that pocket can bind. And so this data is not perfectly conclusive, but we see a larger change in the nano-DSF profile for the Hexapro fold-on in the presence of linoleic acid than we do for the repack9 equivalent, which appears to be consistent with the spike losing the ability to bind linoleic acid, although admittedly not perfectly conclusive. We've also analyzed the repack9 Hexapro fold-on spike by EM, showing that it is indeed a spike. So what's interesting from that is that these mutations appear unique to helping the stability of the isolated RBD and are not as helpful for the full context of the spike. So going back to the isolated RBD context, we then took these trimers that I was previously discussing and assembled them with a complementary pentamer to make the nanoparticles displaying the RBDs, either the wild type RBD or these 2 stabilized RBDs. And one thing about these nanoparticles displaying the wild type RBD is that because the RBD is pretty hydrophobic, the nanoparticles are kind of displaying a lot of different sticky things and can become more sticky and prone to aggregation. To get around this, we've had to approach this with a decent amount of formulation work. And one of our kind of like on-the-go buffers that we've kind of used for just like moving research along quickly involves just glycerol, chaps detergent, and arginine, which is a pretty intensive combination of different excipients. So what we did is we were curious to see what happens if we initially prepared these different particles in this buffer and then kind of removed different excipients and saw how these different stabilized nanoparticle immunogens perform. Shown here is aggregation is assessed by UV-Vis spectroscopy, in this case with higher signal meaning more aggregation. And what we see is that when we remove the chaps detergent, you see more aggregation from the wild type version than you do from the stabilized version. And when you take out arginine, you see still more aggregation from the wild type version, with like the repack9 stabilized version showing less aggregation, showing that the stabilizing mutations appear to help that. We also see similar better maintenance of antigenicity, and that as you remove excipients from this and then measure how well they still bind to like ACE2 or CR3022, you see that the antigenicity of the antigen is better maintained by these stabilized versions, and all showing that kind of from a manufacturability perspective, these stabilizing mutations could be helpful for kind of widening the range of possible buffers that these could be amenable to, which could be particularly useful as vaccines are needing to be developed fairly quickly. Importantly, we wanted to analyze what the immunogenicity of these all these molecules looked at, and we set up an immunogenicity study in mice with MinFam at our organization handling animal immunizations, pseudovirus neutralization handled by Kate Crawford in the Bloom lab for a lentivirus system, and Lexi Walls in the Wiesler lab for an MLV-based system, and ELISA's handled by Carla Louise Herpold at our institute as well. We compared these 3 nanoparticle immunogens to Hexapro fold-on to prefusion spike trimer without the repack9 mutations as a general comparator, and also compared to kind of try and get a better sensitivity, made versions that were just these trimers of these RBDs alone, but mixed with a version of the pentamer that does not have the interface. This allows us to kind of compare these different groups as like a trimeric immunogen, while also having a direct comparison to the nanoparticles by including an equivalent protein. So shown here are binding titers with a couple different measurements of that, one being area under the curve, which is more sensitive, and the other being midpoint titer, which is less sensitive. Hexapro is shown on the left, and these binding titers are measured against Hexapro, which gives very good responses. The trimeric equivalents are not as initially immunogenic on prime, which measured from week 2. However, the nanoparticles get off to a strong immunogenic start, and the binding difference does get more closed after a second shot. What was interesting is for neutralization, as we've seen before, after a prime, all nanoparticle groups were very potent, while a lot of the trimeric groups were not as potent, and these stabilized mutations didn't, both between the particle groups and within the trimeric groups, the stabilizing mutations did not appear to have any significant impact on immunogenicity. So this is a good thing because these particles are all very immunogenic and give very potent neutralizing responses already. So being able to just have a better, more stable version of the particle that maintains that potent immunogenicity is a very, very good thing. The probably more exciting data that we got at the end of this was from shelf life stability. So these nanoparticles were, in this case, with and without the stabilizing mutations, were stored at a range of different temperatures, with -80 included, 4 degrees, 22 degrees or room temp, and then around 37 degrees studied. And at all of these time points, for 28 days, or all of these lower time points at 28 days, there was no differences observed. However, we did see differences observed at a more extreme time point around 37 degrees, in which, as observed by DLS, the wild type particle started aggregating and lost antigenicity, while the stabilized versions, most particularly our leading candidate repack9, did not aggregate even after being stored for a month at 37 degrees Celsius, and did not lose antigenicity. And this was as well further validated using negative stain electron microscopy, with aggregate formation seen for the wild type and no aggregate formation seen for the stabilized versions. So these, in all, these stabilizing mutations can really help improve the yield and thermal stability of the RBDs, and by changing the antigen's orderness, also reduce hydrophobicity. The stabilizing mutations do not appear to have a significant effect on immunogenicity and maintain the really potent immunogenicity of these vaccines. And as well, they help shelf life stability and solution stability in a way that could be very applicable to vaccine manufacturing. So how is this actually important? So this, we have been exploring these mutations in other ways for future projects, and this is somewhat of a preliminary update, but the data seems pretty clear at this point, in that different RBD antigens from different variants can have lesser yields lesser stabilities and also be more prone to aggregation, particularly in the context of nanoparticles, with the B1351 variant being an RBD that we've noticed for this. So we've found that adding these mutations to, or at least including these mutations in this development process for vaccines using different variants has been important for helping make like adequate amounts of vaccine and avoid any issues with having to change formation or formulation, and also just avoid general aggregation. So at this point, I think these mutations will be most valuable for ensuring that protein-based vaccines that are based on the RBD can be reliably updated as fast as possible, and maximize their scalability and reliability, particularly if that turnaround is needed fairly quickly. And as we are exploring making updated vaccines with these that are updated to various variants, we are at this point including the repac9 mutations in lead candidates for this purpose. So we are so fortunate to have a massive amount of collaborators, and there are so many people that are involved with all the research that was discussed today, particularly from the King Group and our core labs at the Institute for Protein Design. The VISA lab has been a very great collaborator for a lot of structural work and immunogenic analysis, and as well, Bloom Lab has also very much helped with the DMS and also neutralization assays as well. And for many of the other background stuff shown, large thanks to David Baker's group, the groups at the VRC of Masaru Kanekio and Barney Graham, and as well thanks to Ralph Barrick and Bolly Plundren for help with other SARS-CoV-2 studies, and large thanks to our funders, including NIAID, the Open Philanthropy Project, the Bill and Melinda Gates Foundation, and the Audacious Project. And I'm happy to take any questions at this point.
All right, Dan, that was excellent. I've, as always, got a million questions. So audience, please either type your questions into the Q&A box or raise your hand or both. Dan, first I have a question about a more general statement that you made early on. You said that stabilizing glycoprotein antigens is tough. Can you be a little more explicit? Like what's the failure mode there? What happens?
Totally. I think it depends on the type of protein that you're approaching, and I mean in some cases you could make stuff more stable, or you could think you're making something more stable, but really the mutation doesn't quite form right and the molecule might become less stable. As with any proteins, predicting exactly why they lose expression is really hard, and so it's really hard to know exactly why they lose expression. So yeah, it really depends on the exact problem that you're approaching, but in some cases you can infer it, but in a lot of cases, a lot of the modes of failure are pretty mysterious.
Okay, but you would say that this happens more often in your hands for regions that are glycated than regions that are not glycated.
I was more when talking about glycoproteins, more just talking about a lot of the viral antigens that we focus on are glycoproteins, and I mean these are really complex proteins that are do a lot of gymnastics and complex things. And sometimes you can throw a wrench in that in a good way that holds them down in the way you want them to, but sometimes if the wrench doesn't really land right, like they have like, you know, they've evolved to like have instabilities in them. And so it can easily be pushed in the wrong direction too.
We have an audience question now.
Thank you, Dan. Shen, will you please unmute yourself and ask your question? All right, I'll just read out the question. So Shen would like to know if the stabilization mutations, do they affect the antibody response to virus RBD variants, which like CR3022 does not bind to the receptor binding epitope?
Yeah, that's a good question. We're still in the process of studying that, and it's like so far the repack 9 mutations do appear to give equally good responses against variants compared to the wild type version. Are like in terms of like heterologous responses, we do have somewhat of a drop off, but like from our nanoparticle vaccines, but there's no real, as most vaccines do, but we haven't seen any significant differences between the repack 9 mutations and the versions without it.
Good. Dan, got more of a big picture question here. So last week we had Peter Palese talking about the other entry into this landscape. So, but what would you say are what's the killer app for the nanoparticle approach, and how does that stack up against other modular display systems like using a pseudotyped Newcastle disease virus?
Yeah, that the vaccine that they've been working on in collaboration with Jason McClellan's group is super exciting as well. And I think the way I personally view it, which admittedly I'm someone who I'm a biochemist and not the complete expert on it, but the way I personally view it is that we need, in order to get the vaccines for the rest of the world, we need as many different technologies as possible. It's the RNA vaccines have been absolutely great for the US and a lot of the developed world, and have personally changed my life and many other people's lives there who's watching, hopefully. But like there's going to be limitations to getting those to the rest of the world, and there's going to be every type of vaccine out there kind of has its own pros and has its own cons. And in terms of getting all the vaccines that we need to the rest of the world, having the full portfolio of all the different types of vaccines is going to be really, really important. Protein-based vaccines is something that is yet to be fully deployed, but they have a very strong reputation of being very immunogenic and very effective. With the Novavax vaccine being like one leading candidate there. So we hope that at least in the context of protein-based vaccines, the nanoparticles can provide something that is both fairly high yield, which is, the RBD is a very high yield protein. So taking advantage of that's really good. And also using the nanoparticle to ensure that it is a vaccine that's not only high yield, but very high potent. But yeah, we need the full portfolio of everything to get vaccines to the world.
Yeah, no, I agree. But more shots on goal is always better.
Yep.
So while we're on that topic, can you speak a bit to things that are completely out of my realm of expertise of how easy would it be to manufacture, I don't know, let's say 2 billion doses of your vaccine candidates and package them and cost and
Yeah, man, I could speculate, but yeah, I admit I don't know a lot of the precise details about that level of manufacturing to know what are all the pieces that would make or break the difference between 2 billion doses in a timely manner. At least from our side, we've just the things that we've done is we've tried to make this technology as streamlined and easy to handle as possible. And I mean, at least in the context of if we need to do a strain change to a vaccine, having mutations like this that are ready to help out is, that's all we can do. And yeah, I'll hold off from any of the other manufacturing because I just don't know that as much.
I wouldn't be able to evaluate your answer anyway. But okay, coming back more to something that's within both of our wheelhouses. So I don't know if you saw this recent preprint from this Japanese group. It's unsurprising that they observed such antibodies, but so the notion is that, so we all want the neutralizing antibody response where it binds and blocks. We feared antibody dependent enhancement, at least, early in 2020, where it would bind and take it to FC receptor as a backdoor. That seems to be, absolutely not a contributor here. But what this group observed, and I think others have observed, is antibodies isolated from patients that actually enhanced the binding to ACE2. Most of the ones that they found the antigen was actually the N-terminal domain, and it stabilized the RBD in the open conformation and displayed it in a multimeric format to ACE2. But do you guys screen for something like that?
Yeah, we have not been, and to my knowledge, in terms, that's a difficult thing. It's a difficult thing to analyze. The approach that at least the philosophy of at least developing vaccines that are best insured against situations of ADE or other similar things is that I think a lot of us subscribe to is if you're going, like, as long as you have a decent amount of neutralization and neutralizing responses and are able to keep that at a pretty high level, that is something like the ability for like a really adverse event like ADE or something similar to happen is less likely. And I very much say that from like the perspective of like trying to develop a new vaccine, and that's part of one of the reasons why you always follow neutralization and why neutralization is held in like a very high regard, and why people don't just like use like antibody binding titers as like the main thing to follow. So I mean, it's hard to know exactly how responses like that fit into the full context of stuff, at least to my knowledge. But to me, from my knowledge, I feel pretty comfortable that as long as there is sufficient neutralizing responses, such antibodies having a significant impact would likely be lower. Again, speculating a little bit out of like my personal expertise there, but neutralization that's again why we love neutralization.
Yeah, excellent. All right, I've got one last really geeky biophysical question. So I've needed to thermostabilize a ton of proteins in my career as well. It's super easy when they're bacterial, because the oldest trick in the book is you just blast it against thermophiles and steal one of those, and it usually works just fine. We recently managed to pull this off with a fungal protein as well. Those genomes are a bit of a mess, but we managed to pull out the ORFs that we needed from the thermophilic fungi and graft those things. So there are many thermophilic viruses. Every organism on the planet is infected by viruses, including those in extremophilic environments. I did a quick Google search while you were talking about like thermophilic influenza, thermophilic coronavirus. It doesn't seem like there are any close homologs of those guys known from thermophilic environments. But have you guys peeked to see, if there are some areas where you could find thermophilic homologs? Because sometimes you can just glance at it and be like, oh yeah, of course, it's using that proline to stabilize, that little beta turn there.
That's a really fun idea. I've never thought about it that way, but like, yeah, I'm definitely going to Google that. Like, for some of the things that I'm looking at after this, we have taken a similar kind of phenotype guided approach for different projects. One of the, as mentioned, one of the other antigen design projects that I worked on is for influenza neuraminidase. And we found, basically what we found for that is that some families of this protein are naturally more stable than others, and like there are other families that are naturally less stable, and which we validated with our own like phenotypic measurements. And basically then our design process was trying to transfer over like minimal pieces from the stable ones into the not stable ones. So we've approached like, you know, that approach that you're describing is very applicable. But yeah, I never thought about it from looking at thermophilic viruses. And yeah, that's a really fun idea.
Yeah, I mean, I think there's thoughts. I mean, there's all these viruses out there that are incompletely documented and understood. I wonder if like somewhere hiding out there are like thermophilic versions of these viruses that nature already selected for, and we just haven't, you know, they're just sitting in some NCBI sequencing file, just waiting to be mined.
There's just so many viruses in the world that like there's got to be something out there that would match that description.
Yeah. Okay, well, Dan, that was, yeah, thanks for your time. Thanks for your work. That was amazing. Sorry, people, we don't have a closing slide today, but next week's talk is very likely to be at a different time. It is likely to be at 4 p.m. Eastern, but we will have the details on Twitter and on our website. So, but regardless, see everybody next week and stay safe. Okay, thanks again, Dan. All right, bye.